Okay, so we're headed into chapter 6 now, and we're looking here at worksheet number 5. Excuse me. And chapter 6 and worksheet number 5 really mark the beginnings of a new unit. The unit is protein function, and in this unit, we're going to be thinking about functions of proteins such as enzymatic catalysis. We'll think about them as ligand binding proteins. We'll think about allostery. We'll think about cooperativity. We'll think about membrane transport. All things that proteins can do. And of course, we're going to spread that over chapter 6, 7, 8, and 9. Right. In chapter 6, though, we're thinking about enzymatic catalysis, okay, and what enzymes can do specifically to lower activation energy. So at the end of chapter 6, we talked about how enzymes lower activation energy so as to then catalyze a reaction or make a reaction proceed at a much greater rate as compared to the same reaction without the enzyme present, right? And there were several things that we talked about, one of them being that enzymes provide bind energy, the second being that they carry out these specific catalytic mechanisms, and then the third being that they provide these proximity and orientation effects, okay? Now, we're not going to discuss all three of those in this worksheet, but we are going to discuss specific catalytic mechanisms. And the reason is because I have found in my polling of students over the semesters that specific catalytic mechanisms are the subject that confuses students the most with regard to this subject. So I want to spend some time thinking about that, and worksheet number 5 is going to do that, okay? And the very first thing that we're going to be doing in worksheet number 5 is we're going to be drawing out a reaction coordinate diagram, okay? And then we'll start to look at other aspects of the mechanism, specifically like what types of specific catalytic mechanisms we're thinking about here, okay? Now, to do that, we're going to be thinking about this enzymatic mechanism that's sitting here on the screen, okay? And some of you look at this and you might think to yourself, man, that looks a lot like the enzymatic mechanism of GAPDH, or glyceraldehyde 3-phosphate dehydrogenase, and that enzyme catalyzes the sixth step in glycolysis. And if you thought all those things, you're doing great, okay? We're not going to talk about this enzyme until chapter 11, when we'll actually look at the processes of glycolysis, and we'll think about GAPDH in its biological context. But this is indeed GAPDH's mechanism, okay? And it's just one of many, many different mechanisms that we could present, all right, in an attempt to try and understand and help you understand what are specific catalytic mechanisms and what is the purpose of those things, okay? And generally speaking, what we want to think about with those is that specific catalytic mechanisms take a single, very improbable step of a reaction and break it down into more probable steps, okay? And they do this through these chemical strategies that they can employ, okay? Now, in order to draw out a reaction coordinate diagram, you really probably need a little bit of orientation on what you're looking at here. So let me do this first of all, okay? And that is this. I'm going to go ahead and draw just a shape to try and indicate here that what you're looking at here, okay? And so we could just draw something like this. Could have been a variety of different shapes, okay? But what I just drew there, that would be the enzyme, okay? And what you see here then, I'm going to highlight this in red, okay? What you see there, that's the substrate, substrate 1. The name of the substrate is glyceraldehyde 3-phosphate. And so we are then beginning here in our enzymatic mechanism at ES, okay, or ES1 would be another way to think about it. In other words, the enzyme substrate complex is already formed, okay. Now, when you look at this then, okay, and let me go ahead and point to an arrow with an arrow. When you look at that arrow right there, that is something that the enzyme is going to do to induce some change in the substrate. Okay, and that would be essentially step number 1, which is indicated that change is going to take place with his arrow. Okay, so that when we get over here, we have seen the results of this arrow here. I'll put a star by it, that arrow here. Okay, and we're at what would be considered then an intermediate form. Okay, now I could draw this blue outline over here, but it gets really kind of busy if I were to do that. Okay, but I'll go ahead and draw this. Okay, and that is, I'll go ahead and put this here, right? This red outline, that would indicate what is no longer the substrate because something's happened to it, but this would be the intermediate. Okay, now in addition to this, and this is something that you have kind of keep in mind here, okay, with this enzyme, and that is that step one, as it transitions, the enzyme also binds something that we call coenzyme, NAD+, and that would be shown right here. Okay, and you could just call this coenzyme 1. Okay, and then what we're going to be looking at here would be this series of arrows. Okay, so there would be an arrow right there, there would be one right there, there would be one right there, right? And I can just keep pointing, but there are all these little arrows. And again, the enzyme is going to do something that's going to cause a transition, and that's going to be step 2, and it's going to move in this direction. Okay, all right. And in this process, okay, there's going to be another coenzyme that binds. Okay, so here would be another coenzyme, and we could call that coenzyme 2. Okay, and at the same time, you see here that there's a product that's released in ADH, and so we could call that product 1. Okay, and it's released in this second step. Okay, and then of course we have the results of this, and I'll draw this red arrow or red box around this. This would be, right, another one of our intermediates. Okay, it's no longer substrate any longer because the enzyme has done a couple of things now to change that. So it's this intermediate form moving towards what we would consider product. Okay, now again, we could look at arrows, right? So there's another arrow right there. The enzyme and its coenzymes are going to do something, okay, that's going to have induced this third change, okay? And in this third change, then we can put a box around this, and we go like that, right? Right, so this is another form that the enzyme takes, and then finally, that thing's going to be released as product 2, okay? So we could call that our second product, okay? All right, so there's a lot of nomenclature there. The nomenclature I don't want you to get too hung up on, but I went ahead and told you about it, right? But what I want you to do is take all that information and draw a reaction coordinate diagram based off of this. Now, if you haven't attempted that, then go ahead and pause it, do the reaction coordinate diagram, step number 1 of our worksheet, and then come back and listen to the feedback. If you have already, but you're like, oh, that makes a lot more sense based off of what I was doing, go ahead and maybe redraw that, okay? And then we'll bring this back and we'll think about it, okay? All right. Okay, so I trust that you have drawn your reaction coordinate diagram at this point in time and that you're ready for some feedback. You're eager for feedback. What does this look like? Okay, so let's go ahead and look at this. And how would we draw a reaction coordinate diagram for this? So reaction coordinate diagram, let me go ahead and draw the axes for this. Have that axis here, the y-axis is G, and then we'll draw here the coordinate, the reaction coordinate, we'll just call that RC, how the reaction progresses. Okay, and so when you come back up here, like I said, we're starting in the ES, but in our reaction coordinate diagram, we want to model, okay, the formation of the ES. Okay, so maybe you didn't do that, and you're like, oh no, now I have to redo it again. So pause it, redo it, and come back and see what you've got here, okay? But let me go ahead and proceed on. So what we want to say here is our very first step, and this would be true that any enzyme is going to be to go from E plus S to ES. Okay? And that's going to be the first step. Now we start somewhere else, okay, beside that, but that's where we would go first, okay? Now, step number 1 then would be up here, right? Indicated by this arrow here, and that's going to lead to our, our first intermediate, okay? So I'm going to go ahead and draw this up here. Now, I'm going to go ahead and put a star here, and I'm also going to put plus C1, or actually not plus C1, I'm sorry, just C1. Okay, C1. Oops. And that's to indicate that we have bound this in the same process. Okay. Now, when we look at this, and we have to go back to our instructions here. It says, write out a reaction coordinate diagram for this reaction, assuming that step 1, the step after ES formation, is rate limiting step. Okay, so that's a very important aspect of our instructions. Let me go down here so you can see this. Okay, and that's going to help us understand what our activation energies are. Okay, so there's step 1 after ES formation. Okay, now step 2, I'm going to go ahead and do this. I'll go ahead and draw something that looks like this. I'll put it about right here. ES, and we'll call it star star. Okay, and then I'm going to put the product. Okay, so I'm going to say, actually I'm going to put C2 because we have to have our coenzyme come up here. We're adding our coenzyme. But then I'm going to put plus P1 because we release product there. Okay. Okay. And then the next thing that I'm going to do here is draw another one. And this would be step number 3. Okay. So we're to right here now. Okay. And I'm going to put EP because we're forming that second product. And then the final thing that we're going to do here is going to be for our E plus P. And it's really E plus P2. Okay. Because it's our second product. Okay. And we're releasing P2. Right? So those are the steps of our enzyme. Now, some of you are thinking to yourself, man, mine looks so much different than yours. Okay. And that actually is going to be okay. Okay, because there's some freedom in the way you draw this. Some things we have to have the same, but other things we don't. Okay, but let me just go ahead and do this. I'll just draw the landscape here. So we'll go like this. Then we'll go like this. Then we'll go like this. And then we'll go like this. And then we'll go like this. Okay, so that's our landscape. Okay, now, what are things that we need to have the same? Well, first of all, you would need to have this ES depressed in free energy relative to E plus S. Okay, in other words, the ES is a more stable form of the enzyme and substrate than if they're separated. Okay, and that's because of the binding energy that occurs between the enzyme and the substrate. Okay, now, the instructions said that then the first step after ES formation is a rate limiting step. Okay, so what we have here, this would be, we'll go ahead and use different color here. What we'd have from here to all the way up to here, that would be our transition state. Okay, there. And this would be then, that arrow that I drew would be the activation energy. Okay, and I'm going to call that cat. Okay. Now, I've had some people ask me about this in office hours and everything. Maybe you've seen this as capital E subscript A activation energy, and that will be okay as well. We're going to use delta G double dagger cat. Okay, but that's our activation energy. Okay, and so we say that this activation energy is the largest of all the other activation energies within the reaction coordinate diagram because it's the rate limiting step of our reaction. Okay, now we could say this, okay, and think this is important. We could look here and say, well, this is an activation energy, and this is a transition state. It's shorter than the other ones. We could say that this from here to here is an activation energy, and we could say that this from here to here is an activation energy, and each of these would be transition states. Okay, so we have several different transition states. Okay, but the one that has the largest activation energy between essentially the preceding form, in this case ES, to that transition state, that's going to be a rate limiting step, the step that's the slowest, and therefore it has the largest activation energy, and it will also have, incidentally, the smallest rate constant. Okay, all these other steps here, here, and here are activation energies, but those are going to have really large rate constants. They're very probable events. Okay, all right, so having ES depressed compared to E plus S, and having this, our first step, being the largest activation energy, those things need to be consistent across all of our reaction coordinate diagrams. Now, some of you might be saying, but I put this here, and I didn't use that nomenclature, ES star star C2 plus P1. I put it up here. I put it higher than ES star C1, or the previous thing. And so you might have something that looks like this. And is that okay? You might be thinking to yourself, you didn't draw it that way, so it must not be okay. And I would say, actually, it is okay. And the reason is because you don't have any information on the free energy of that intermediate. So you don't know if that intermediate is higher in free energy than the previous one or lower in free energy than the previous one. I put it as lower, but it could be higher. Okay, either one is fine. So long as our activation energy, when we draw it out, doesn't exceed the activation energy of the rate limiting step. Okay, and the same thing could be said here. Right now, when we look at this, E plus P2, okay, when we look at that, there is some constraints on that. We couldn't, for instance, put it here. And the reason that we couldn't put it there is because we know that our reaction, when we look at it, it has a positive delta G at standard conditions, which means that the free energy of E plus P2 has to be higher than E plus S. Okay, so that would be true. You'd have to keep that consistent. Okay. All right. So other than that, you have to have this number of steps, and it has to take on a form like this. Okay. But there is some freedom in the way we've drawn this based off of the information that's given to you in the worksheet. Okay. All right. Hopefully that makes sense. Now, some of you are saying, that's great, but how does that really help me out? I see that, I understand the reaction coordinate diagram. What does this all mean? Right. Well, this goes in the direction of helping you understand, okay, the purpose of what we call specific catalytic mechanisms. Okay, so if you imagine the reaction without the enzyme, so let me just go ahead and cross this off. Okay, and what you would have to envision here is that the reaction is going to take place like this. Both products are going to be made at once, and it's going to go directly from S to P, and it's going to skip all this stuff in between. In other words, there's going to be a single hill, okay, and it's going to go off the screen, it's going to go way up there, and then it's going to come way back down, okay, and that would be the uncatalyzed step. There's only a single hill. Okay, and you can imagine as it goes off the screen that it's going to reach some peak, and there's going to be then some activation energy that's going to, end off the screen. Okay, and this would be our delta G double dagger, and I'm going to call it uncatalyzed. And it's going to be really large. Okay, and what we've done here is we removed all those intermediates because the chemical strategies are not present. The enzyme provides the chemical strategies to break that one very improbable event where S becomes two different products, okay? Not to mention, the NADH that's coming off and that kind of stuff. Well, actually, that was one of our products, sorry. Okay, but it's going to produce those all in a single step, okay? And like I said at the very beginning of this recording, that specific catalytic mechanisms are going to be something that takes a single, very improbable event and breaks it down into multiple, more probable events. And that's what we're showing here, is that each one of these intermediates are the result of a chemical strategy that we call specific catalytic mechanisms that broke down that one large event into several individual steps that are more probable. So come back up here, right, and we can highlight for you, right, step 1, and step 2 and step 3, okay? And we can look at those and say, oh, those are specific catalytic mechanisms. The enzyme is employing these chemical strategies to take a very improbable event and make it more probable because it breaks it down into several small digestible steps, okay? Right, and by doing so, then it lowers activation energy. It's essentially a new path to product, okay? Right, so to try and use an analogy again, if I were to go from Fort Collins to Greeley or Fort Collins to Denver or something, there's several paths. In fact, there's a large number of paths that I could take. Okay, for instance, I could go to Denver through Los Angeles, right? Meaning drive all the way to Los Angeles and then drive all the way to Denver. Well, that's going to take a really long time, but it's a path. Okay, it's one path, or I could just hop on I-25 and go straight down. Some of you say, I don't know, I think I'd rather go to LA. I think that I-25 traffic will take me just as long. Okay, it won't, but I get the joke. Okay, but in any case, that's a different path. All right, and one of them is improbable. You're not going to go to Los Angeles to go to Denver, right? The other one's more probable. Okay, and that's essentially what we have here with specific catalytic mechanism is there's a more probable path, and it has these little steps in it. Okay. Okay, now, what we need to do next is think about, well, what are these steps? What are these chemical strategies? And to do that, we need to name them. Are they covalent catalysis? Are they acid-base catalysis? Are they metal-ion catalysis? What are they? Okay, and so step 2 says, in our worksheet, says, identify what type of specific catalytic mechanism step 1 and 2 are. Be prepared to answer poll everywhere question on this. Obviously, we're not going to do poll everywhere in this environment, okay, but you get the idea. So let me go ahead and pause it for you. Or not pause, I'm not going to pause it, but ask you to pause it, and think about, well, what are step 1 and 2, or what are they showing? And let me go ahead and erase all this jazz. Okay, jazz is a great word to use for all that. Okay, you got rid of all the jazz now, and what we're going to think about is this jazz. Okay, what does that arrow mean? Okay, because that's going to induce step 1, and then what does this arrow specifically mean? And that's going to induce a whole nother set of arrows, and that's going to be step 2. Okay, and it's going to induce, step 1 is going to induce the formation of this intermediate, what we call the thiohemiacetal intermediate, and this step here is going to produce what's called the thioester intermediate. Okay, and those are specific terms. Thiohemiacetal and thioester are specific to this enzyme. Okay, not every enzyme is going to do this, but what we need to think about is what do those arrows represent? Do they represent covalent catalysis? Do they represent acid-base catalysis? Do they represent metal-ion catalysis? Right, so think about that, okay, and name them. And specifically, not just acid-base catalysis, is it acid catalysis or is it base catalysis, right? What is what, okay? All right, so pause it, think about it, and then come back and we'll look at this. Okay, so you're back now. Hopefully you pause it, you thought about it, you named them, okay? And so with this one, let's look at number 1. What we have here is the cysteine side chain with that sulfur group on it. It is a deprotonated sulfur, which means it has a negative charge, and it is a nucleophile. Okay, and nucleophiles are going to then attack electrophiles, and that carbonyl carbon on, it's actually an aldehyde, okay, but that carbon on that aldehyde is really electrophilic, okay, meaning that it can be attacked by a nucleophile, and that's exactly what happens here, okay, and what we see here, the result of that is going to be then this, a covalent bond between the sulfur and the carbon, which means that this is what we would call covalent catalysis, okay, a nucleophilic attack of the substrate, the enzyme being the nucleophile, okay. Now, the second one here, okay, that arrow, this one right here, right, what is that illustrating? Well, in this case, what we see here is that, I'm going to go ahead and focus in on this a little bit. There we go. What we see here in this is that this nitrogen is going to take that proton off of the intermediate, okay? Meaning that our enzyme, in particular histidine 176 in our enzyme, is acting as a proton acceptor, which means that it is a, all of you are saying, base. Okay, so this would be base catalysis. Now, the result of that is the rest of these arrows, right? So this is what the enzyme does right here. And then the result of taking that proton is that these electrons go here, and 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 that makes this thing called NADH. And this is actually ultimately a redox step, okay? We'll get into that later on. We don't need to think about redox chemistry right now, but what we do need to think about is, okay, that step right there is a base catalysis step, okay? Now, some of you are saying, well, why aren't these metal ion catalysis? And the reason is because none of these have a metal ion on it, okay? In fact, GAPDH doesn't require a metal ion, okay? Okay, all right, now, the last question here, come back to this, okay? It says, finally, answer the following questions. What would happen to the activation energy of step 1 if cysteine 149 were mutated to an alanine? Would this cause the rate constant to become larger or smaller for this reaction? Okay, so think about that. Okay, in fact, let me do this. Let's think about this, cysteine 1499. Instead of it looking like that, it's going to look like this. E. Okay, that's the enzyme. And coming off of that is going to be a methyl group. All right, so if I put a methyl group there instead of that sulfur, that negatively charged sulfur, what is that going to do to the rate constant in my first step? Is it going to increase? Is it going to decrease? And why? Okay. Pause it. Think about it. Okay, so hopefully you've come back now, and hopefully you pause it and you thought about it, and the rate constant of this is going to get quite a bit smaller. And the reason is because that methyl group on alanine, so this would be an alanine mutation, is not going to readily attack the carbonyl carbon. It's not very nucleophilic at all. And so it's very improbable that you'll get a nucleophilic attack of a methyl group to that electrophile, that aldehyde. And therefore a rate constant would be quite small, and the activation energy would be quite large. And in fact, this enzyme, if that mutation were to occur, would no longer catalyze this reaction. Okay, all right, so that would be very detrimental, of course. Now it says change cysteine 149 to serine. What would this do to rate constant? And I'm going to be honest with you, this one's a little bit tricky. Okay, so because what we're now doing is this. Serine is going to look like this. The end of serine will be this, OH, and you think, oh, that oxygen there, that can be an electrophile, can it? It can be, but that proton would have to be removed. And the question would be, could that proton be removed for that oxygen to act as an electrophile? Okay, and so I'm going to say this, that's a tricky one, and it's debatable what would happen. I would be more inclined to think I would have to do an experiment on this, okay? Could the enzyme still catalyze the reaction at a similar, if not advanced rate, with the serine there instead of the cysteine? Okay, my hunch is that it won't, okay, because I don't think there's a way for the enzyme to get rid of the hydrogen off of the hydroxyl on the serine, but it's hard to say without running the experiment. So, okay, right now, some of you are saying, well, man, that's hard. Why would you do that to us? Oh, only to make you think about it. Okay, I wouldn't do that to you on exam because this is fairly ambiguous as to what would happen. Okay, I would rather put you in a more unambiguous situation like the mutation in alanine. Okay, so I'm going to go ahead and stop our recording right there. I think that's plenty good. Hopefully you have learned a little bit more about what it means to have a specific catalytic mechanism, how that helps an enzyme carry out its reaction, okay, and what they are, these chemical strategies, and how to identify some of them. Okay, right.